Again, good evening, everyone. For those who are just joining in now, we'll start now. On behalf of SESTA, Archivia, and BioVentures for Global Health, BVGH, I want to welcome you to this week's session of the training. This week's theme revolves around data management. So we are going to start off immediately. And the first presenter we have for this night is under the auspices of Archivia. He's an Associate Director of Quality Management at Archivia for Sub-Saharan Africa. He's by name Dr. Maxwell Anyango. You're welcome, Dr. Anyango. And I'll please urge the admin to share his slides so that he can start his presentation. Once again, welcome everyone, and please ensure that you fill out the attendance if you've not done so. Dr. Anyango.
Thank you very much, and good evening, everyone. Please confirm you can hear me loud and clear.
Yes, we can.
Thank you. So I've already been introduced, so I will go ahead to the presentation. So today we're going to discuss general principles on data management. It's a very wide topic, so we might not be able to cover all the details in the 30 minutes time. But for those who like to get more information on what it entails in terms of data management, you can refer to good clinical data management practices. It's a huge document, over 500 pages, publicly available online, so you can check it out, and you can always refer to it for more details. So we can move on to the next slide. So when it comes to data is the most valuable currency when it comes to clinical trial implementation. And why do we say so? It's because all the activities, all the things we've been discussing for the past 1 month or so eventually leads to data. And it is that data that will eventually be analyzed to tell us if our trial has met its objectives. And out of that, then the decisions will be made as to confirm whether our investigational product is working or it is not working, and if patients eventually will get the new drugs or not. And therefore, it is critical that when we generate data out of a clinical trial, then we ensure that that data is accurate, it is verifiable, and it is credible, because if it meets those criteria, then we can have the confidence, the public can have the confidence, and the regulators, that the decisions resulting out of that data are sound. And therefore, it means that as investigators who are conducting the trial, as sponsors who are funding the trials, then they have a huge responsibility to ensure the data integrity, to ensure its traceability, and most importantly, to ensure its privacy. So in the next few slides, we're going to discuss some of these principles in a little bit more details to understand what it entails in terms of practical implementation of clinical trials. So we can move on to the next slide. So we will start with data privacy. And as all of us are aware, in the recent few years, there's been a lot of discussion and talk and regulations surrounding data privacy, both at the international space and even within our local jurisdictions. So when we talk about data privacy, then we are simply referring to standards for protecting personal data. And personal data is any data that singly or in combination can be used to identify a unique individual. And these are just a few examples of what constitutes personal data, but the list is much more larger than that. So any of these details here that can be used to identify a unique individual constitute personal data, and therefore the data privacy laws and regulations apply. And therefore, when we are conducting clinical trials, we always want to remember that there is that element of data privacy, which now is regulated more stringently than a few years before, before the EU started by enacting these laws, and then almost all the countries around the world followed suit in enacting laws to govern how data privacy should be handled. So we can move on to the next slide. So, what are the minimum standards when you are handling, collecting, processing, and even storing personal data? One, it's important to train institutions to have a policy that governs how personal data will be handled so that we prevent privacy incidents and also avoid potential regulatory penalties that might result as a result of data breaches. Then the other aspect that data privacy plays a part is when we are designing our CRFs or source document templates. It's important to use as minimal subject identifiers as possible so that we only include personal data where it's absolutely necessary or where there is a justifiable business need for collecting that personal data. And if not, then we are good to just use, unique subject IDs. And if we must have a link log where we collect the personal data, like the full names and date of birth and telephone numbers and things like that, then we store that separately in a place that is access controlled. And when we are designing these CRFs, it's important that we remember personal data will apply to all those documents that we will be using and processing in the course of that clinical trial. The other aspect where we need to be mindful of processing personal data is when we are transferring that data, especially when it is moving out of site to either a sponsor, a medical monitor somewhere, or any other person who is not based on site, then it is important to ensure that we do not submit that information, including personal identifiers. So we will need to have a process or a policy in place that we blank out completely any personal identifiers whenever source documents or CRFs are being transferred out of site. Then have a process in place where on a regular basis we review our site processes to ensure that they are always in compliance with the current privacy laws and regulations. Because again, in Africa, we live in a jurisdiction where this regulatory environment is constantly changing and new laws and policies might be revised or introduced at any given moment. So when we move on to the next slide, then what are the best practices? So when we're handling personal data, of course, the participants, we have to seek their consent. They have the right to share this information or not. And they have the right to be informed on how this information, this private information will be processed and used and for what purposes and by whom. And most of the time, this information is captured in the informed consent form. But we've also seen a few instances where the template of the informed consent form does not capture this. And at times, the reason behind that is because either the institution or the ethics committees are using templates that have always traditionally been in use before some of these laws were enacted. And after they were enacted, those templates were not updated. and therefore it makes them non-compliant now when being reviewed in line or in the light of these new policies. So it's important to review even our informed consent templates to ensure that they conform to data privacy laws that might exist within our jurisdiction. But most importantly, at the institutional level, we need to foster a culture that is privacy conscious so that we are sensitive on how we handle, how we collect and process this information, and even how we store it. Then it's important that we have pre-transfer procedures in place so that in the event that we need to move that data, we do not find ourselves in a situation where we create a privacy incident because, for example, the site has shared with the sponsor photocopies of source documentations that contain personal identifiable information, because the sponsors are very sensitive to receiving that information, simply because in some of the countries where they're domiciled, they have very strict laws that govern how that kind of information is used. And therefore, in the event where that information is inadvertently shared with them, then usually it raises a lot of issues. So it is a best practice as a site to develop a policy so that everyone is trained and aware on what to do in the event that personal identifiable information needs to be transferred out of the site. And therefore, for accountability purposes, it means that we need to have a process in place. So if not a policy that's already existing, then we might need to create an SOP that governs how personal identifiable information will be handled in the course of conducting a clinical trial. Then, of course, it is not always possible to absolutely exclude processing of personal data, because for as much as we assign subject IDs to participants, but still we need to collect their names, we need to link it up with their source documents which have their subject IDs, and therefore for whatever reasons, at times it necessitates that we have personal identifiable information. So then how do we keep it? It's important that we have a process in place that defines how that personal identifiable information will be stored on site and who will have access to it. And then also it's important to have a policy in place on how the site will process requests for access of personal data. So these requests might come either from the sponsor or third-party vendors and things like that. So by default, usually it should be to deny access for that. But in the event that there is legitimate business reason for a third-party vendor or a sponsor accessing that, then we must have a process in place that conforms to the local laws that will not put us at loggerheads with the regulators. Then, of course, security measures, whether we are storing it in paper format or in physical forms. So like those identifiable information should be kept separately with the other CRFs. Some sites develop that process under lock and key and have, a log that traces who has access to which folder for what particular purpose and when was it returned and so on, so that you have an audit trail at any given time to be able to know who is accessing personal identifiable information and so on. If you are keeping it in electronic format, then we want to have passwords and firewalls and all those electronic security measures that the IT people are familiar with to ensure that it is not accessible to anyone. Next slide. So then again, now we want to discuss about case report forms, because that is another practical aspect that as sites will be involved in designing for purposes of collecting the data that is generated during the conduct of a clinical trial. And ICH GCP Revision 3 now has expanded the definition of the CRFs to include whatever format that is being used, because for as much as traditionally paper has been used, but of course with the adoption of technology and so on, now we have all sorts of digital formats and media that can constitute source documentation. So the principles are the same irrespective of the media that is being used to collect this information. Now, the most important thing is that it must align with the study protocol so that we ensure all the data is collected that will be required for assessment of the efficacy endpoints and the safety endpoints. And therefore, if we do not design the CRFs properly, then it means that the possibility of it compromising the study outcomes is very high, because at times you might only realize when you want to do analysis that, oh, we did not collect certain variables. And usually at that time, it is late, and even attempts to try and collect that information retrospectively only can introduce recall bias and raise other queries and question marks as to its credibility. And therefore, it's important that when we are designing the CRFs, we must understand the protocol very well. And we are going to look at the more details in terms of how to go about doing that. But what are the components of a well-designed CRF? One, it must simplify the database design. So we want to ensure that our CRFs are not overly complicated, because if they're overly complicated, if they collect too much information over and above what is needed for the purposes of the study, one, it increases the administrative tasks, because for those who are involved in clinical trials will appreciate the amount of paperwork and data that is involved. So we want to limit it to what is essential for the protocol and what is essential for reconstructing the events around the conduct of the clinical trial. Next slide. So what are the best practices when it comes to CRF design and data collection? Now, one of the best practices is it's important to have standardization. So when you are conducting several studies at the site, the best practice is to maintain a library of standard CRFs that you can just customize to fit the protocol-specific requirements, as compared to developing new templates every time you have a new study. And the reason for that is because most of the time you will find that it is the same site staff who are involved in running these different protocols. And therefore, if there is no standardized CRFs and each protocol has its own design and format, then it becomes very difficult from a user's perspective to correctly fill these protocols, because most of the time they tend to run concurrently. But if you have a standard way of collecting certain information and certain forms are the same, then it becomes even easier from the user perspective. And most of these variables will always be the same, because the demographics will always be the same, vital signs will always be the same, physical exams and things like that. So it's important to have a standardized templates, so that you only customize the few points that are protocol specific. Then the other best practice is to have a collaborative methodology when it comes to the design of these templates, because when you have input from the data entry people, from the statisticians, from the clinical staff, then everyone is able to make their contribution in terms of how they interact with these forms, as opposed to just assigning one individual to develop all the templates. Because if, for example, you just give it to the data manager to develop the CRFs, they may not have the details in terms of what is the participants' flow within the clinic, and how does that information move from what procedure to what procedure. And therefore, if it's only one person developing it, as I have observed, At times, the templates do not come out as they are supposed to, or they do not meet all the study uh, requirements, even in terms of meeting Alcoa principles, simply because it did not factor in what is the participant's flow or pathway within the clinic, because the data person will only look at it in terms of what is the EDC requiring, and those are the data points that he will transfer into the CRF. Then the other thing is you want to look at the endpoint focus. So what does that mean? You want to ask yourself, what are the endpoints for the study, and what data points will be analyzed to meet those endpoints, and how are you going to capture them? So when you do that, then you will ensure that you have captured all the data points that are required for the purposes of uh, meeting the objectives of the study, and there are no parameters that are inadvertently missed out. The other thing also is to look at it from the end user perspective. So, for example, if you look at the EDC and you're simply using the EDC template to generate your source templates, at times you might miss the fine details from the protocol. So let me give you an example. So you're designing a CRF and you're capturing the vital signs page. The EDC will simply ask what were the vitals, what was the blood pressure, what was the respiratory rate, and things like that. However, if you look at the protocol, it might have said the patient must rest for 5 minutes or 15 minutes before any of those vital signs is taken. So that duration of resting will not be captured in the EDC system. However, in your source templates, for a monitoring perspective and from a compliance perspective, you should be able to demonstrate that this participant indeed rested. And therefore, it means that detail must be captured in your CRF design. So it's always important to look at the protocol, one, and then two, look at what is the flow. The other example I might give where we see a lot of gaps when sites design these templates is the physical exam page. So physical exam, we know in practice will also include the vital signs. But we know in practice, most often than not, it is the study nurses who will take the vitals, then the clinicians will do the physical exam. But in the EDC, this might be captured in one page. But in the clinic, these two procedures will be done by two different sets of teams. So the vital signs by nurses. So if you just copy and paste the EDC design the way it is and translate it to your templates, then what we see most of the time is there's only one signature page at the bottom. So now the nurse, because they took the vital signs, they're not able to complete their initial and date to say they're the ones who took it, because there's only one signature page at the bottom of the page, and therefore it is left for the clinician. So now what happens? They are now forced to write it in pieces of paper, and then they hand it over to the clinician who will transcribe it into the ECRF. Now, once you adopt that process, now the transcription for the clinicians, it's no longer source, because the source is what the nurses took and recorded initially. But if there is a collaborative approach, then the nurses will be able to contribute and say, it is us who take the vital, and therefore we also need our space to include our initials and date. Then the other thing that you need to factor in is to try and, as much as possible, to minimize redundancies and referential data. So when you are designing it, try as much as possible to have one source for one data point, so that you do not have the same data point recorded so many times within your CRFs or transcribed so many times, because every time we introduce transcription, we are increasing chances for errors and inconsistencies. Next slide. So as you design these CRFs, again, the other thing you want to ensure is that every single page must be attributed to a particular subject. So the subject identifiers must be up there. Then the unique identification of the different separate sections. So a good CRF design will have sections, as opposed to it being like a booklet where you complete it from page 1 or 5. Okay, because if you develop it like that, then it becomes difficult to standardize it and be able to adopt it across protocols. Then it's always important to remember that every site staff needs to document whatever they did in a contemporaneous manner. And therefore, if it follows the participants' flow within the clinic, then you'll be able to know this procedure is different from this other one and it's done by a different person. And therefore, at the end of it, I need to include sections for initials and dates for the person conducting it. Okay. Then when you are thinking of the layout, again, we said, think about the end user, think about what process is done first, what follows next, and then from there, where does it move next, so that you avoid it, the users flipping from page 1, then it needs to be completed page 5, then they go back to page 3. That way it flows smoothly and everyone is able to complete it as they complete those particular procedures. You're able to simplify the wordings and the codings, and even in terms of performing quality controls, they're able to easily see that the person who just saw this patient has not done ABCD, and therefore it minimizes the potentials for errors. Next slide. So let's also again discuss about performing QC of these CRFs, because this is one of the essential elements that contribute to good quality data. And even in terms of delegation, what you have seen at times is sites completely forget even to delegate who will be performing QC of the data that is being generated at site. And I've seen that generating inspection findings and audit findings and so on. So remember that quality control activities are part and parcel of the process of generating good quality data, and therefore you must always have personnel dedicated to perform these activities, and you should incorporate into the designs of the CRF how they are going to document their QC activities there, so that it is independently verifiable by any person conducting audits or inspections. So when you're performing QC1, you want to confirm accuracy and completeness of all the sections that needs to be completed. And 2, you want to check for any missing data that either has been omitted for errors of omission or commission. It's important that when you're doing that QC, please interact with that information and see what is it telling you. Because at times when QC staff are performing QC, they are simply looking for missing blanks and unticked boxes and so on without even reading that information. Because remember, the purpose of you looking at it is to reconstruct the events for that day and see, does it make sense? Does this data make sense? And therefore, if you don't read it, you'll not be able to pick the inconsistencies. You'll not be able to pick things that do not make sense. So if, for example, you're just reviewing the vital signs field, you'll be able to say, okay, the temperature has been completed, but they have recorded the temperature is 32 degrees. It does not make sense. Or the temperature could be 39 degrees. Yeah, have they indicated clinical significance? Has an adverse event been reported and so on? So interact with that data and see, does it tell the complete story and is it consistent? Then in terms of timeliness, want to confirm that these CRFs are completed in a timely manner. So different studies will have different timelines, but generally, usually they're within 2 to 5 days of the participant's visit on site. So any data that is being entered into the system past 5 working days is already late entries, And of course, at times it can happen because the site recruited very fast and there's a lot of data backlog. But it's important that at the site level, the PI continuously assesses this situation so that They confirm, is it an issue of staffing and the data staff needs to be increased, or is it an issue of technology, internet, and things like that, so that as much as possible, we can stick to these timelines for data entry. Then, of course, legibility and clarity. If there is any writing that is not legible, please try as much as possible to ensure that if they are handwritten, everyone can be able to write it. Try to avoid the use of ambiguous abbreviations, because well, in clinical trials, people love using abbreviations. For me, you must have heard I kept on saying EDC. I was assuming everyone knows what that is. But we want to remember that different abbreviations might mean completely different things for different audiences and even in different regions. So unless it is a standard abbreviation that is recognized across the industry, try as much as possible to write them in full. Then if there are any changes or corrections that need to be made, please ensure that the site staff who did that has initialed and dated, and the original entries have not been obscured. And at times, it might be necessary to provide clarifications as to the rationale for making those changes, especially if when the change was made and when the original entry was done, there's been a long time lag that has happened. Next slide. Then, in terms of audit trails, it's important also, again, to maintain audit trails in terms of making those changes and so on. Then, compliance with the guidelines. So when you are entering the data, the sponsor usually will issue the CRF completion guidelines that will guide the site in terms of how that data should be entered in the electronic data capture. So please ensure that those who are tasked with that responsibility have reviewed these guidelines and ensure that you have them. And if the sponsor has not provided them, please request for them, because that is what will help you to know how to enter the data into the system. And also it will standardize how different sites are entering the same data. Because if there is no SRF completion guidelines, then what happens is there'll be a lot of variability in terms of how people are interpreting that data and how they're entering it into the system. And during data cleaning, it will generate a lot of queries. Then confirm that the investigators or the site staffs who are entering that data, they have signed off wherever they're supposed to do that. And for the electronic data capture, usually it is designed in such a way that after the data has been cleaned, then the investigator will have to sign the CRFs, and usually they can also delegate one of the sub-Is to help them in doing that. Then finally, in terms of storage, confirm that where we are keeping these files is secure, is a controlled environment, is safe from premature destruction of these source documents, because we've had incidences where files disappeared, and it becomes extremely difficult to try and trace where the file disappeared to, and it becomes a huge privacy incident, because if a file has disappeared, then we don't know who has access to that information, and at times it does contain personal identifiable information. And it also means that the information in the EDC now is no longer usable because it is not backed by source. So it's extremely important that we review the policy surrounding how the files are stored on site, both temporarily when the study is ongoing, and then we think about the long-term archival, because there are regulations and rules surrounding how long should this be kept. Next slide. So then let's quickly talk about laboratory data again. So the principles surrounding the handling of clinical data apply also to laboratory data. And laboratory data is equally important because for most studies, it constitutes the data that will be used for analysis of safety endpoints. And even for some studies, depending on the nature of the protocol, The efficacy endpoints also might be obtained through laboratory data. And we have different types of laboratory data. It could be those that are for efficacy endpoints, safety endpoints, and or even for specialty data. And for specialty data, we're talking about things like, genomic studies, PK, pharmacodynamics, pharmacokinetics, genetic markers, and things like that. And this data might be generated from different labs. So we might have central labs where sites are simply collecting the samples and shipping it to a central lab where all the results are analyzed and the results are shipped back to the different sites. Or you could be processing it at your local lab within your facility or in specialized lab for things like pharmacokinetic and pharmacodynamics and genetic markers and so on. So each of these setups have their own advantages and disadvantages, which we will not go into the details of discussing for now. What I've seen is there's a lot of trajectory towards use of central labs, and simply because it standardizes how those samples are analyzed and the assays are analyzed, and then the results are generated from a standardized, similar, assessments, as opposed to local labs where every site might be having their own process and assays and things like that, which introduces a lot of variabilities. But then there are other studies again that you must just use local labs, because the advantage is that the results come much faster as compared to the use of central labs. Next slide. So again, best practices, like we talked about for clinical data standardization, again, is very important. For lab data specifically, reference ranges is extremely important. So before the study begins, even the sponsor will normally request for what are your local reference ranges. So ensure that your reference ranges are specific to the trial population that you want to study, because it is based on the reference ranges is what will determine whether the results are flagged as abnormal or not abnormal. And then you'll be required to, assess the clinical significance and so on. Then in terms of data transfer and review and reconciliation, the principles are the same as for the clinical data. Next slide. And in terms of unit conversion, as much as possible, try to use the actual units that will be required for the purposes of analyzing that data set, because normally when you resort to doing manual conversion, then there's usually the chance for errors that come in. And therefore, as a best practice, try as much as possible to use the SI unit for that particular lab value. Avoid as much as possible doing the conversions, though there are certain parameters that you might just have to convert or to manually calculate, because the labs do not provide you with the final values. Then if there are any errors, because at times the errors might occur, for lab data, usually the corrections are done using the lab correction forms or DCFs. So ensure that if that is the established process for the study, then that form is completed and then this is sent to the central lab. Then they'll be able to change that data from the back end. But if it is at your local lab, then at times it might be much easier to change that, because it will not be in an electronic format. But the ones done at the central lab, usually they're already in an electronic format, and they're simply pulled into the database. And therefore, if there are any changes that need to be made, then you will need to complete that data correction form. Next slide. So I like to discuss assessment of lab reports again, very important when it comes to lab data. So your responsibility as an investigator will be to receive the report, whether it is from the local lab or the central lab, then assess for clinical significance of any out-of-range results. And you need to assess for each and every out-of-range results and see whether it is clinically significant or not clinically significant and document it in that form. So most protocols will say if it is assessed as clinically significant, then they have to be reported as adverse events. Okay, so as part of the procedures will be confirm that there is consistency. So once you say it is clinically significant, then look at the adverse event log and confirm it has been completed there. Okay, then do your initials and dating. And when you're doing your QC, those are the things you'll be checking for the lab results. And if for any reasons they missed one or two parameters, then you flag them and ask them to reassess it and document on that form, whether it is CS or NCS. And then the information will be entered into the database. Next slide. Okay, that's my last slide. So I'd like to pause there and take any questions. I think I saw some questions in the chat.
Thank you very much, Dr. Yango. Thank you for the presentation. Please, I will plead that you hold on while we take the second and last presentation, and then we can address any questions during the Q&A session afterwards.
Okay.
But you could look up some of the questions that have already been posted on the chat so that you could prepare ahead. So thank you very much. So we're continuing on with the presentation, still on the theme of data management. And we're going to be listening to something that has to do with, that relates to us, especially since there are a lot of us from the SparkNet sites here. We'll be listening to Mr. Ruben Chianumba, who will be taking us through registry data, especially using SparkNet or Sparko in terms of site readiness, data handling, and data quality in clinical trial readiness. So please join me and welcome Mr. Ruben Chianumba for his presentation. Over to you, Mr. Chianumba.
Thank you so much, Prof, for the warm introduction. I want to also appreciate the previous speaker, Dr. Nyango, for his excellent presentation. He had at least helped me, to reduce my slides. While he was speaking, especially with regards to data quality, I was quickly deleting some of the slides that were no longer necessary. So, just like Professor Maxwell said, I'll be talking about the SparkNet registry in site readiness, data handling, data quality in clinical trials. I hope that at the end of this presentation that I'll be able to explain the importance of the data management in clinical trials as it relates to SparkNet registry, describe the SparkNet registry and its function, and then discuss how the registry enhances site readiness for clinical trials, examine data quality control processes, and then maybe identify a few challenges while, based on the experience that we have on the SparkNet. So why data quality matters in clinical trial? Dr. Nyango mentioned a lot of things about data quality issues, how important that we manage data accurately. He mentioned that data is the currency for, in fact, every research activity, including clinical trials. So accurate, timely, and complete data are the backbone for regulatory approval and patient safety. Clinical trials fail, in my perspective, not because of lack of the intervention efficacy, but also due to poor data quality and management lapses. So for me, I feel that if you manage your data well, the data outcome or the outcome generally for that study improves. So for the SparkNet registry, just to throw a little bit light on our SparkNet registry, a Sickle Pan-African Research Consortium Nigerian Network, SparkNet, It's a multi-site registry, 25 SAD clinical sites across the 6 geopolitical zones in Nigeria. The SparkNet registry is centralized in University of Abuja. It's a cloud-based database designed to collect, manage, and harmonize patient data across multiple sites. The registry is integrated and it's integrated in, and as I mentioned, it's a cloud-based data system designed to collect clinical, demographic, and laboratory information on individuals with sickle cell disease. So the registry is hosted in the instance of the University of Abuja, now Yakubu Gowon University. So the map on the right shows a clearer picture of the distribution of the sites across the 6 geopolitical zones in Nigeria. And the wonderful faces you're seeing in the picture are the site leads across all those sites. The slide below showing the distribution of SS, S-beta thalassemia, and SC patients in the registry. So with this picture alone, you can say, okay, this is the number of SS patients, S-beta thalassemia patients, and the number of SC patients in our registry. And as at September reports, we have a total of 10,000. We had a total of 10,574 patients enrolled in the database. But so far this month, we have over 10,600 patients enrolled in the database. Yeah, so the table showing the age category male and female is just to show us the distribution at enrollment of patients in this different age category. And you can see the male and female population too. So one can simply say, oh, how many male and female patients do you have in this age category that can be screened for a possible clinical trial enrollment? So the purpose of the SparkNet registry is to establish harmonized data standards. Now, these data standards is not only in the SparkNet Nigeria registry, but across the Sparkle consortium, all the participating countries. So we have a standardized case report form collecting data across all sites. So it facilitates patient tracking, follow-up, and reporting. So our database can help facilitate the patient tracking, follow-up, and reporting. There was this instance during the clinic visit. A patient reported to Professor Nodu that her weight, she has been attending the clinic here over a period of 2 years, and then her weight has been reducing. She was weighing 60 something, and then Prof requested for her clinical information. In that patient's case, throughout the entire period she has been attending the clinic in our facility, we've been recording not just her weight, her height, other clinical information. And when she made that report, Prof was able to pull out those information and say, yes, we can be able to map out and report, okay, this is what the patient has been going through, or this is what the picture of the patient's I will say history. Yeah, that's the word I'm looking for. So the registry helps us to be able to, at any point in time, understand the true picture of the patient and in real time ascertain if we have this number of patients with this category to be able to be enrolled for a clinical trial. And then it also provides baseline data for clinical trials. So if a clinical research organization wants to start up a clinical trial in our site, they will give us a checklist, eligibility checklist to say, let us know using this inclusion and exclusion criteria if we have patients eligible for a particular study. The SparkNet registry also strengthens data infrastructure and human capacity for multi-site trial. With regards to site readiness and capacity building, one of the key roles of SparkNet is enhancing site readiness. Before a clinical trial can begin in your site, sponsors, ethics committee need assurance that a site has one, adequate patient population. So you must show that you have adequate patient population. You must show that you have trained personnel to be able to carry out that clinical trial. And then you must show functional data management systems. In our case, we have, apart from the hospital electronic medical record system, we also have the red cap registry, where we can say give a full picture of the patients that we have we are enrolling in a clinical trial. Now we also have a network. We call them Warriors Hangouts, where patients interact and they discuss issues concerning their well-being. In that platform too, you can able to say that most of the people there are not only in the registry, but have one-on-one interaction with the health professionals. So for you to be ready for clinical trial participation, you must show functional data management system. You must show established quality assurance processes. Okay, so for our registry, we have these layers of proof that we are ready for clinical trial, where our data is available, our data is reliable, our data is usable, and is ready for use at any time. So how does registry support trial feasibility? I know I was showing us a chart in the previous slide where you can see the quantity of SS, SC, beta thalassemia patients. We can also check their treatment history. If they're using hydroxyurea, if they're using folic acid, their clinical manifestation, how many crises do they have in the past 12 months? Most clinical trial organizations will ask you, can we know if they've had at least 2 to 5 VOCs in the past 12 months? Can we know if they've had blood transfusion? Can we know if they have so our database is able to pull up that information. Okay. Then follow-up compliance. How many follow-up does that patient have? We also can be able to pull that up from the database. Beyond that, this partner strengthens human capacity, for trainings and participation across sites and for other studies, possible studies. So the SPACNI registries identify eligible patients population, as I mentioned, predict recruitment potential, demonstrate existing data governance structure. For instance, using the SPACNet matrix, you can estimate the sample size of a possible SCD drug trial using some of those things I mentioned. I kind of, called this from a clinical trial investigator that wants to assess if we have patients, that can participate in the study. So I've kind of tweaked it a little bit for this presentation. I titled this eligibility criteria for ACAM study. The inclusion criteria includes male, it can be a male or female, between the ages of 12 and above, confirmed diagnosis of sickle cell disease. Now, there's this reoccurring incidents that if you come to the clinic, most patients will tell you I'm SS, but they don't really know if they are SS or H beta thalassemia or even SC. So some study investigators will want to know documented SCD patients' genotype. So these are some of the informations that will be included in the inclusion criteria if the patient has a documented SCD genotype information. If it doesn't, then, before the screening, that patient should have one. Have between 2 to 15 episodes of documented VOC. Now, this is the inclusion criteria from the clinical trial investigation organization. They want to make sure that patient that's going to be enrolled for this study has a HB of above 5.0, HB equal to or above 5.0 and less than or equal to 10.0 gram per DL. So these are the inclusion criteria. And I've also learned over the years that if you meet all the inclusion criteria, you may not be included in the study because the exclusion criteria may also exclude you from that study. So these are some of the exclusion criteria listed here. Use of voxylator or similar agent within 28 days, use of selectin antagonist, receiving regular scheduled blood transfusion, and that is about maybe above 6 or equal to 6 transfusion events in the past 12 months. Participants who have received RBC transfusion for any reason, receiving or using concomitant medications that are strong inducers of this protein, and severe renal dysfunction, and maybe a travel distance on standardized 6 minutes walk test. So some patients may not be able to take this test, or are not able to do a particular, or take this, 6 minutes walk at screening. Those people should be excluded from the study. So these are the inclusion and exclusion criteria. So from our SparkNet database, we're able to, before the screening date, pre-screen these patients. So we can be able to list out 10, 15, 20 patients who are eligible based on your inclusion and exclusion criteria. Those are some of the benefits that the SparkNet registry, helps in clinical trial. So for data handling, and data management workflow. We will now look at how data handling works maybe in practice. The first chart you're seeing shows the 4 stages of data management, at least from our SparkNet registry. The first is the data capture. Then you move to data validation, data transfer, and then the review, monitoring, and feedback. So these are the 4 stages. For data capture, data is first captured on a standardized case report form and directly moved into REDCap. So we have an electronic form. Previously it was a hard copy. Now we have REDCap. We're using the mobile app for enrollment, and we do that real time. So these forms are aligned with the SADC data dictionary to ensure that all participating countries collect comparable variables. So apart from the fact that the Nigerian sites have common variables that we collect across all the 25 sites, this data is also comparable with the countries participating in the Sparkle. So one can easily analyze the data across those sites and then, give a proper information about the patients. Validation. Built-in validation for us, we use some rules in REDCap, such as skip patterns, logic branching. So if you say that you're a female or you're a male, questions relating to females will pop up. So we won't ask you questions that are associated with the females only. So those kind of ones. And then if you say you don't smoke, for instance, questions following smoking and then habits of smokers will no longer pop up for you. So those kind of branching logic, we use them to validate our REDCap. Then for transfer, once we have validated the database, we have ensured that the data is clean, accurate, and transferable. We've removed all the identifiers, just like Dr. Anyango has mentioned. You have to remove all identifiers before you transfer data. We back up and then we move it to a centralized database at SADC. Okay, for us in Nigeria, our database is hosted in University of Abuja, and we back it up in the cloud and also the hardware. Then review and cleaning. So we do different levels of review. We do review in our sites, and we also do review at the SADC. So SADC will also look at the data and give you a feedback, providing you with a data quality certificate. In that certificate, they expect to see how complete your data is, if there are outliers. Okay, outliers, I mean here, if, for instance, the date of diagnosis of that patient, the date that the patient was diagnosed of being a sickle cell disease, is earlier than his date of birth. So that means you've diagnosed that patient before you gave birth to that patient. So in that situation, it's an outlier. It's wrong. It will be flagged off, and then people are expected to check and correct those things. So SADC look at those data quality issues and issue a certificate based on your performance. So some of the key steps in our data handling and then management workflow in our sites include data capture, verification, validation, transfer, and review, just like I mentioned. So this is also a snapshot of our REDCap. I just wanted to mention here that our data is harmonized with the SADC. At least 92 variables are expected to be collected across all participating countries, including Nigeria, alongside other variables that your site may wish, to include. So for us, we have a follow-up form that is unique to our site, incorporating the standardized data variables that is expected from the hub, as well as those that are unique to our own locality. And then we've also modified the database We don't take things like ethnicity, because we don't take ethnicity. We take tribe. No, sorry, we don't take race. Rather, we take tribe. Okay, so yeah, and then we have controlled vocabulary encoding based on the SCDO, that is a sickle cell disease ontology. So the picture you're seeing here is how the red cap looks like for those who are familiar with the platform. For those of us who are not, you have a menu section where you have the project, the red cap information, project setup, the forms, the kind of forms you are collecting. Are you collecting screening form, demographic form, anthropometrics form, follow-up form? And then you can also see other applications like the export function, import function, then the user rights data quality function. So this data quality is where you can now ask how many issues, how many missing data, how many outliers before you can now find the sites where or the participants who has that incomplete information. So I just took a picture of this and modified it a little bit so that we understand for us what we look at when we open up our RedCap database. So for patients 5936649, these are not real patients. The screening is complete, demographic is complete. However, the anthropometric, which include maybe the height, weight, is not complete. And that is why it's in red. And then if you see them in yellow, it's unverified, meaning that perhaps the data assistant has filled the information, but it's not sure. Just like Dr. Yango mentioned, that you don't just enter data without asking questions. So if a patient, for instance, has a temperature of 90 degrees, you should be able to ask question. How can somebody have that kind of, it may be a typo error, okay? And if somebody has a PCV of 1, one should be able to ask questions. Why is this value? So some data assistants, research assistants, DMO, notice some of these challenges and then they keep them in amber color, amber or yellow color. And it means that these are unverified. And then if you look at the column where the follow-up information is, you can see patients who have multiple follow-up informations appear in this color. However, those who don't have follow-up information can just see just one. So for this patient, perhaps he just came for screening, he doesn't have any other information. This could be a newborn, and then again, it could be many other issues. It could be a data issue. So we have to ask questions. Okay, so I'll just move faster. Yeah, for data quality, there are different dimensions we look at it. We look at it from the angle of accuracy. How accurate is your data? Okay, we look at it from the angle of completeness, just like Dr. Yango also mentioned, timeliness, consistency, and then the integrity. Okay, so how accurate is your data? Does it represent the patient's information in reality? I mentioned a patient who came and complained about her weight. And then when we went back, we found out that over the years, truly, this patient has reduced in weight. So does it really reflect that patient's information? When we click this information of this patient, do we find out if this patient is a pediatric patient with all the informations available for that patient correctly written? Okay, can we go back to if, for instance, you have a paper-based case report, that is hospital case report form, and we go back and see if those informations tally with what is there? Is it accurate? Okay, then we'll go to completeness. Is that information of that patient complete? Did you just take a patient's date of birth? What of the gender? What of the year of diagnosis? What of the SCD type? Okay, then timeliness. How early? He mentioned 2 to 5 working days. For us, the standard is 48 hours. You have to make sure that you enter them promptly so that you don't even forget. Okay, then consistency. How logical across variables is this? I'll give us an instance. So we're taking heights in centimeters. Okay, and then as a data person or as a site lead, the red cap instrument has shown that you're supposed to take the data in centimeters, and then you go ahead to take them in meters, and every other person is recording 170 centimeters and you are recording 1.7. Okay, those are some of the challenges or consistency issues. One should make sure that you're following the units, as properly as possible. So for us, we ensure that when we do quality checks, that people are consistent, people maintain the same pattern in entering them. The date of birth must match. We're using the same type of date of birth. So we validate those cells. You can't take our date of birth and maybe year, month. No, you have to take them day, month, and year in a particular format. So the integrity, is it maintained during transfer? So when you're transferring the data, whether within the sites, among the sites, or to the hub, how do you maintain the data integrity? So these are some of the things. But in all, data quality ensures that accuracy, completeness, timeliness, consistency, auditability too, all play a role to make sure that this is a reliable and trustworthy data. So some of the data quality issues that we have encountered is for our SpeakNet database is duplicate records. So we'll find out that some patients, and this may be occur when you have multiple data management officers entering the record, or you employed a new one. Maybe you change record research assistants often, and then when they see patients, and then some of these patients, if you while enrolling them, you give them stipends or you give them some benefit, maybe medication, or you run some lab tests for them free, there's a possibility that when they come for their next clinical appointment, that seeing a new face, they'll tell the person that they are a new patient, that they are coming to the clinic for the first time. And then maybe the research assistant or whoever is, consenting them for the enrollment, just because of the benefits, they will lie, and then you enroll them. So sometimes they don't go back to check the information. And then when we now do kind of audits, we find out that there are some patients who have been repeated in the database. So some checks has been put in place to make sure that these patients, that this occurrence doesn't happen. Another common issue we experience in our registry and sometimes is inconsistent patient identifiers. So RedCap generates record ID. You can't do that yourself. And then the site is expected to put in their hospital record number. That's the hospital number. Yes. So you're expected to put in the hospital number. So we found out that over the years that we've managed this data, that some sites, that the pattern, of the hospital number changes, maybe with a different administration, or maybe a change of system. If you are using a paper system and then you move to an electronic system or something, the record ID pattern changes. And then those patients who have, who had that, maybe they have moved them into new record ID system, and then the one that on the what do you call them, on the red cap becomes different. So your new enrollments will now have patients with a different pattern of a medical hospital number. So these are some of the occurrences we see. Some sites will just suddenly start having new record ID, medical hospital number. I'll use that word hospital number so that don't confuse them. So they use different hospital number, the pattern over the years. So these are some of the things. Then missing values. Yes, this has, in fact, I don't think we can stop talking about missing values in data entry. People always find it convenient to skip information that they don't have. I think it's better than feeling what you're not sure of. However, we try as much as possible to avoid missing this. There are some informations that are not supposed to be missing. For instance, a patient's gender. You cannot afford to miss such kind of information. Patient date of birth. These are very, very important information in our RedCap database. We found at missingness in these areas, especially demographics, and then as much as possible some of the clinical information. In fact, if you ask me to say, I would say that all informations on the electronic or paper-based case report form should be filled completely. Okay, then there are some out of range values. For instance, somebody born in the year 1905 and is still available, is still alive. Sometimes we find that that can occur as a result of typo error. And then again, just like I mentioned earlier, people who have their date of diagnosis, the day that they diagnosed them of sickle cell disease, coming earlier than their date of birth. So those are some of the challenges, data quality issues that we see. And then late data entry. Some people will enter, so they enroll the patient on 1st of January 2025, and then they are entering the record a month later. Between that time and when you enroll that patient in on RedCap, many other sites would have enrolled patients on that RedCap. Okay. And then the record ID assigned to that patient will come later. However, you enroll the patient earlier. You see, so by enrolling the patient late, you have distorted the record ID numbering that RedCap does. People who now came after the patient will now be given a record ID, and then you now see the database having information of patients who have enrolled in different times, appearing earlier or later. Okay, then this example I have here is a patient record. Maybe a clinic, a research assistant recorded a patient as 18 years old in the first visit with a height of 150 centimeter, and then the next visit, which is visit 2, just 3 months later, the patient now becomes a height of 170 centimeters. So when we are looking at this kind of data quality issues, we tend to ask question: is it possible that a patient will grow from 150 centimeter to 170 centimeter in a week or 2 weeks? Or is it that you did not get the right height the first visit? The gap is too much, and we tend to ask questions. So this is just an example of some of the data quality issues that we have in RedCap. Okay, just to continue on this data quality assurance framework. The SparkNet registry operates under 3 tiers of data quality assurance framework. First off is the preventive quality control. So it's achieved through training, standardization, SOPs, and automated system checks in RedCap. So this is how we achieve the preventive one. So we try as much as possible to validate some cells so that you don't enter them wrongly. And then we have a standardized CRF, standardized SOP, and the system check to prevent you from making those errors. And then we have the detective quality control. And this one is the regular audits. We present our data reports every Monday. And then at the end of the month, we prepare a data report for the steering committee. So within those periods, we look at the data completeness. We look at the data issues that are available, whether you have outliers and all not, and then we audit them, and then we kind of give a feedback. If there are issues that need to be corrected, we reach out to the research assistants. Then the last but not the least in the data quality assurance framework is the corrective quality control. So if we have noticed that you've entered data wrongly, maybe you've entered data that does not fit to the age of that patient, we give a feedback, and then we also do a retraining. We regularly do training and retraining, and then we do data re-entry where necessary. So if at enrollment you have those informations wrong, and then there's no way we could quickly find a way of correcting the errors there, that they're just too much, you may have to re-enter them. So data quality is not the end point. However, it's an ongoing process. So for us, we try as much as possible to plan, do, check, and then adjust. And then I will just quickly mention the role of a data manager and the site team. So data manager also involves data entry people, data entry clerk, the DMOs, both in clinical trial and even in our research database. So we have an SOP, and it's edited by version. Currently, we are on version 3.0. Effective date 19th of February 2025, and due review date 18th of February 2027. Okay, the site PIs, data managers, and the ITMS director are those involved in reviewing our SOP. And this SOP is standardized for all CESTA researches. So if it is SparkNet, if it is Accelerate, if it is The malaria study, whichever study we are doing, the SOP is standardized for all sister activities. So data manager ensures adherence to data entry SOP, timely upload and queries resolution, coordination of registry support. So, and then the site PI ensures that data accuracy and oversight is ensured, making sure that the data people and the site leads understand the importance of the study. Therefore, regular trainings are very, very important. So I kind of tried to see if I could just make a case study using the registry data for trial preparation so that, I mean, if you are asking what kind of studies can be done in your site with your registry or your information, this is an example for you. So a site used the SparkNet data to identify 350 eligible patients for hydroxyurea adherence. Okay, so how did they come about the 350? They screened the database using the inclusion and exclusion criteria, then identified eligible participants for the hydroxyurea adherence study. Perhaps they may say they want patients who are hydroxyurea naive. They may want patients who have started hydroxyurea and then maybe stopped, and as the case may be, pull out those patients, and then you check for completeness. Okay, the registry tracking revealed about maybe 92% retention and 85% data completeness. Okay, so 92% of these patients that we picked have 350 of these eligible patients have 92% retention, meaning that they have follow-up informations. They've shown 85% data completeness. That means that perhaps all their demographic informations are complete, including address, phone numbers. Those ones can be added in the study. And then this is evidence in the site. This is some things that we can count as evidence for site readiness for clinical trial. So lessons learned for us in the SparkNet registry. The registry provides a data backbone for clinical trials. Quality begins at the point of capture. We insist that at least all data clerks, DMOs, the point of capture, ensure that you capture what is. Regular staff training is essential. And then regular audits to prevent data issues. So it's important that at least monthly that your data is audited and your quality issues are checked, so that maybe when you now want to use the data, you won't have too many data issues. And then continuous communication between the registry, the study investigator, and data managers, how we have found is very important to ensuring that the registry is up to date, useful, reliable, accountable. In summary, the SparkNet registry is a good foundation for all trial readiness. Our data quality is top-notch, it's trustworthy with every evidence of accountability and reliability. And then it's very robust because it encompasses people of different age and then different tribe and then ethnicity and religious affiliation. Thank you so much. I hope that this presentation was very useful to us. I will stop here to take questions. Thank you.
Thank you very much, Mr. Chanumba, Ruben. Thank you so much. That's a very practical approach. So we'll quickly take questions now. So we'll allow you to catch your breath and then also look at the chat like Dr. Yango has done to see any questions there. And we'll call you up shortly. So Dr. Yango, please, some of the questions that were on the chat, please can you address them now?
Thank you very much. So I saw one that was asking how do we capture that patients rested adequately before taking the vital signs. So when you're designing the CRF to take that data, of course, you will have the field where you will record the vital signs, but you'll also have the field for time of the vital signs. So then you will also need to add another field of the time when the resting started and ended. And then when the monitor will be reviewing the source, they'll be able to see the time difference between when the vital sign was taken and the time when the resting started and ended is within what the protocol is stipulating. So if it is 5 minutes or if it is 10 minutes, then they'll be able to tell that difference. So that is how you will design the CRF to capture those different time points.
Thank you. There was also something else that I saw. It says there's build-up of electronic data. Most centers now are going electronic, and hard copy forms are starting to be obsolete more or less. So how would you do an SDV with electronic data without hard copies? That was a question that was on the chat.
Yes, I'm saying it now. Yes, so SDV will still be done because SDV is simply comparing the source date from the source document and then versus what has been entered into the EDC. Okay, so it means you will be having two electronic systems, one which is the source where the sites enter that information, and then you'll be having the electronic data capture like RedCap that has just been demonstrated now. So you'll be comparing the screen from the electronic source against the screen in the electronic data capture. That is how you perform the SDV. The advantage of using electronic source is that it captures the audit trail of who imputed the data and at what time point it was imputed, and therefore the issues of manually signing at the end of your entry do not apply. But then again, it also depends on the design of that. If there are any changes, it will capture it electronically the way normally it captures in EDC. So there are certain advantages of using electronic source data, but SDV is still possible because I've seen sites that have adopted that and it still works. From a monitoring perspective, the approach is slightly different from paper, but it's still possible to do SDV.
Thank you, Dr. Anyango. Just to add to that, sometimes, in fact, not sometimes, oftentimes, if a site or a hospital is using electronic monitoring registry or electronic medical registry, for instance, you hardly will find those hospitals allowing for access to people who are not eligible to have that access. So, for instance, if you're not a doctor working there, you won't be able to have the access. So, if you're coming as a site monitor or something, you're not going to have that access. So, most times you will still have to recourse to like printing out documents in terms of hard copy for site monitoring visits. Now, just to add a question to this pattern, if you have someone, for instance, a participant who comes in and the person, for some reason, has an SAE, hospitalization, right? And this is a hospital where everything has gone electronic. And part of what will be required is that there should be source documents to substantiate that hospitalization, right? Now, if you've done any lab tests, for instance, because that person came in as a patient of that hospital, their details will be clearly on the lab results if you need to print them out, which you will have to if there is no access given to outsiders for the electronic medical records in that place. So when you print it out, you have to do a lot of redaction, covering the names, covering the age, covering everything and all that. How would I don't know, sometimes when you do that level of redaction in order to remove all the identifiers, obvious identifiers. How do you still prove that that result belongs to that participant? Assuming you're asked, because you're going to submit all those documents have to be uploaded either to EDC or whatever. The values can be put in EDC electronically, but when there's site monitoring, you have to actually cite those documents as per source documents.
Mm-hmm. That's a good question, and it's important to clarify here at this forum, because you see there is the aspect of privacy and confidentiality. But then again, you also noticed during our presentation, we both mentioned the issue of data transfer. So when you talk about personal identifiers, I mentioned that at times it might not be possible to exclude the use of personal identifiers completely. So in the instance that you've mentioned, these are medical records. They contain personal identifiable information. Personal identifiable information resides or domiciles at site. And therefore, the site can process and keep and store that information on site. The need for redaction only comes when that information needs to leave the site. And the reason for that is because we do not need other people who are away from the site to be able to link that information to this particular participant. But I always say when participants come to the site knows their names. The site calls them by name. The site does not call them by their participant numbers. They do not say participant number 2, participant. They say this is Okocha, this is so and so. So they know their names because that is how the normal interactions occur at the clinic. And therefore, when now you say you have their medical records and now you redact everything to the extent that now you cannot link it to any participant, now it beats the purpose. Okay? So personal identifiable information can be stored on site, but it needs to be stored in a secure manner. There needs to be processes and policies that govern how that information is handled to avoid privacy incidences, but the site can maintain that for the purposes that you've mentioned, to be able to link all the data to these participants. Because if you redact everything, as you correctly highlighted, there is no way of confirming that this information belongs to this participant. However, if for whatever reason you need to upload it in the EDC or you need to share it with the email to the medical monitor or the sponsor or any other individual who is not on site, then it must be redacted.
Thank you very much. I see that I want to thank you, Dr. Yanga. I also want to thank Dr. Chanumba because he's been addressing the questions on the chat. A hand has been up. I think we'll just take him and then we would close the session. We'll call Professor Nodu if she's to help close the session. So Suleiman, your hand has been up. Please, can you unmute and ask your question or your comment? Please, can the admin unmute Suleiman? It looks like we're having problems with unmuting him. Okay, so let's just take the other person and then Charles Obiora.
Yes, thank you, Prof. Good evening. Thank you for the opportunity, and our lecturers this evening, you've been great. I'm Dr. Yango, the first presenter. While I'm trying to wrap my head around the much you've given us this evening, I actually want to know the extent and the involvement of the CCMW for would-be participants? Of what extent, and how does it encourage their being enrolled in any study too, please? I think you mentioned it in one of the inclusion criteria, please. Thank you, Doc.
Okay, so thank you, Vera, thank you. I think that question is for Mr. Chianumba, but just to say that that particular mention that he did of the seed meaning work has to do with a particular study. Did you understand? It has to do with a particular trial. It's something specific to a particular trial. Okay? It's not as if it's and of course it has to do with participant eligibility now.
Professor Maxwell, you
Yes.
cannot be closing the session when I had a question on the chat and Oyangosi hasn't answered it. So the question is about, I think it has to do with clinical significance, right? Where in a population, I want him to talk about clarify clinical significance of out of range values in a patient population who do not have normal values.
Okay.
You understand? You remember we had that issue at our side. I just couldn't get myself around it. You know, so I want you to explain.
Okay,
Yes.
so let me explain.
Yes.
So
Thank you.
remember, when it comes to assessment of clinical significance, usually it is left to the investigator's judgment or opinion to be able to interpret those results in the context of the pathology or the disease indication under study. So, for example, let me use sickle cell disease as an example. We know that participants who have sickle cell disease in steady state, their normal hemoglobin levels will usually be below the ranges of the normal population who do not have sickle cell disease. So for the same HV value, for a non-sickler population or for a non-sickle cell disease population, it will be regarded as either moderate or even in some instances severe anemia. Whereas in a sickle cell disease population, that will be considered as normal because of the disease state. And therefore, depending on the indication under study, you will find that the normal reference ranges for a sickle cell disease population that is under study for certain HB value parameters, when it comes to assessment of clinical significance, the investigator will assess them as not clinically significant due to the underlying pathology or the disease state. Whereas if it was a non-sickle cell disease population, then it will become clinically significant and therefore it will have to be reported as either moderate or severe anemia. So when you are performing that assessment, you have to use your knowledge of medicine and of the disease under study to be able to determine whether we are reporting this as clinically significant or not. Because once you say it is clinically significant, then it means an adverse event has to be reported, which eventually will find itself in the safety database. And that is why the population reference ranges is extremely important when we are talking about lab data and ensuring that we have accurate reference ranges that is reflective of the population under investigation. I don't know if I have addressed your question.
Thank you, Dr. Ayango. Suleiman, we've not been able to unmute you, or you've not been able to unmute yourself. So I had requested that you type in your question in the chat we will address later. This is past beyond our time slot. I want to thank our presenters, and then I want to invite the Director of Ancestor, Professor Nodu, to close up the meeting. Thank you.
Yeah, so I want to thank everyone for the opportunity to join. I have noticed a declining number, of participants, and I also noticed that some people are coming in towards the end. Just to reiterate, this is clinical trial training. Is to be taken very seriously. Certificates will only be given to people who attended completely. So please let us take this very seriously. And I want to commend everyone for the interaction. I want to thank our guest speaker, Kevin Oyango. We want to thank you so much for the excellent presentations. And I also want to thank Ruben for making data management issues very clear. I'll recommend every SparkNet site to get that video and then play it continuously to your research assistants so that they'll be able to have an insight. There are some questions that people are asking about RedCap, which I don't think were answered, but because of the shortness of time, we'll not be able to go into that. But RedCap is for institutions. Is institutions that sign up for RedCap, and then certain studies are based on it. It's a free software, is in use globally. You don't have to pay for it, but you cannot own it as a person. Your institution will have to sign up for it because there are certain data management issues that go on when people are using RedCap. So that is what we want to say for now. Please get the presentations and go to impact. Today's lectures are just excellent and worth listening to over and over again. All the videos are at the CESTA website, and you can go in there and then listen, until you're familiar with what we are talking about. I want to thank everyone, and then I wish you a good night's rest. Thanks to the resource persons for the excellent presentations. Thank you very much. Thank you.
Thank you. And
Yes,
good night.
Maxwell. Yes, thank you. Thank you very much. Good
Good night.